Hello everyone, I'm MASAKI in Research Institute of Green Science and Technology, Shizuoka University. Here, I would like to introduce our study entitled Behaviors of Plasmids in Different Environments. This is my self-introduction slide. My major research field is plasmid biology, including environmental microbiology, molecular genetics, and microbial ecology. My recent research topics are behaviors of plasmids in microbes, evolution and ecology of plasmids in nature, co-evolution of host bacteria and plasmids. Today, I would like to briefly talk about the first one, behaviors of plasmids in different environments. First, I'll give you a little background. Plasmid, my research target, is an extra chromosomal element in a bacterial cell. While chromosomal DNA is indispensable for the host survival, the plasmid DNA is dispensable. Several plasmids can be transferred among different bacteria by conjugated transfer from donor strain to recipient strain. The recipient strain that obtained the plasmid is called the transconjugant. Plasmid transfer is a central process in microbial activities that control our health and the environment and is a promising tool for their improvement. So it is definitely important for promotion of bacterial rapid evolution and adaptation. Also, the plasmids are essential tools for molecular genetics. Importantly, the plasmid behaviors are involved in the occurrence of multi-drug resistant microbes and their spreading. Therefore, the objective of our study is to understand the plasmid behaviors in various environments. For understanding the behaviors of plasmids, our questions can be divided into three. First, which plasmids are transferred in microbial community of different environments? When and where? Who transfers to whom? How frequently they are transferred? To understand which plasmids are transferred, we did plasmid capturing from microbial communities in different environments. Then, their whole nucleotide sequences were determined, and we successfully obtained more than 50 new plasmids. In addition, the distribution of these newly found plasmids were investigated, and some of them were widely distributed in geographically different sites in Japan. These facts indicate that there are many overlooked but important plasmids which can spread different genes in nature. As for understanding who transferred the plasmid to whom, green fluorescence proteins and a cell sorter are used. After mixing the donor with the GFP genes on the plasmid with microbial communities, cells with plasmid were detected, separated by the cell sorter. Then the cells were identified to know which bacteria can be received the plasmids, and then we compare the host ranges of different plasmids. This information is very important to predict how the plasmid can be spread in nature. As for the frequency of plasmid transfers, we compare the conjugation frequency of plasmids in different conditions. Here, I show an example of our recent study, the transfer frequency in different concentration of oxygen, aerobic or anaerobic conditions. As you can see here, the conjugation frequency of plasmid was lower under anaerobic conditions. Based on these questions, we had obtained several important aspects. Here are the lists of papers published in these 5 years. If you are interested in our research, please read them or contact me. That's all. Thank you for your watching.